Okay, we're going to get started, so try to ignore the fuss at the door. I guess they will let those that didn't register in after 5 minutes. So we do need to get started because we have a full session and we want to stay on time. So this session is Interdisciplinary Metabolomic Epidemiology, the Pathway to Clinical Translation. So I just want to quickly say there was a little bit of confusion. In some places it said interdisciplinary, and some places it said multidisciplinary throughout, whether it was the agenda or the app. We're not sure what happened. But as you will see, this session is going to focus. We are going to talk a little bit about multidisciplinary research and describe it, but the ultimate thing is to show how we've gone from multidisciplinary research to interdisciplinary research in the field and what that looks like. So first, the objectives are to describe the important roles that distinct domain experts play in the process from discovery to clinical translation in metabolomic epidemiology. We want to identify important areas where crosstalk between domain experts is currently lacking, and we want to identify specific steps that can be taken to better establish an interdisciplinary team for metabolomic epidemiology research that will promote moving initial discovery to clinical translation. So, when we started to think about this session, we wanted to make sure that people understood the difference between multidisciplinary and interdisciplinary research. I'm not sure that it's always clear because we use these words, we throw them around. Sometimes we use them interchangeably, and they shouldn't be used interchangeably. So I'm going to take a minute to explain the difference between the two so that as we move into the rest of the session, when our speakers are talking about, when they're saying multidisciplinary or they're saying interdisciplinary, you all understand exactly what they're talking about. So for multidisciplinary research, it means different disciplines that work on a research question independently. So if you see in the diagram, we have, our epidemiologist, our clinician, our biochemist. They're each working on the same problem, but sort of in their own lane. And then they have shared research goals, but they examine the question from their own discipline's perspective, as shown in the diagram. And then each aspect can be analyzed by a particular discipline, which is often necessary to answer complex research questions. So the findings are supplementary to each other. So like I said, they're all staying in their own lane, they're working, they're sort of doing what they need to do, and then ultimately, those discoveries, through looking at the literature or as things kind of progress on the line, eventually they're consolidated into like a more major discovery. And as you can see, that's how it works. So you go from the consolidation of these individual things into a major discovery. And importantly, multidisciplinary research is the building block of interdisciplinary research. So I'm going to quickly describe what interdisciplinary research is, which does differ from multidisciplinary research. Now, I usually don't like to put this many words on a slide, but I feel like this is important because the National Academies, they had a report facilitating interdisciplinary research, and they actually defined it. And sorry, can you make the slides bigger on here? I'm having trouble seeing it, and I have my bifocals on, so everything's blurry, so my apologies. This is what happens when you get older. Thank you, much, much better. I can actually see now. So I usually don't like to put this many this many words on a slide, but in this case I felt it was important. So they define it as a mode of research by teams or individuals that integrates, okay? I'm underlining the important words, integrates information, data, techniques, tools, perspective, concepts, and or theories from two or more disciplines. Integrates two or more disciplines, or bodies of specialized knowledge to advance fundamental understanding to solve problems whose solutions are beyond the scope of a single discipline or area of research practice. So you're integrating between two or more groups to solve a problem beyond the scope of a single discipline or area of research practice. Okay, so that's the difference. In the multidisciplinary, you kind of stay in your own lane. Eventually, people realize you've all been looking at the same problem, and the field ultimately looks at it and kind of consolidates the information. Then you have a major discovery. In this case, if you look at the diagram, you can see everyone's working together on the same problem, same thing, and they make the major discovery as a team. And the unique thing about this is ultimately it is going to potentially end up, as I have down here before, it could lead to a fundamental shift over time, which ultimately leads to a new interdisciplinary field. And that brings me to metabolomic epidemiology. Now, I'm not going to go through this figure in depth. This was a figure from a paper by Jessica and Craig up here, who are speakers, Dave Broadhurst and Rachel Kelly, where there's a epidemiology task group from the Metabolomic Society, where they sort of defined and introduced this new field of metabolomic epidemiology. And this was the figure we used in the paper. So you can pull the paper, but essentially the gist of it is, you're sort of you're working together, you're doing the two things, two or more disciplines. In this case, you're doing everything with epidemiology. You're looking at, all the, sampling, all the things an epidemiologist would do. And the folks in the metabolomics are doing, all the metabolomic, assays and everything kind of together, and they're flowing together. Then they ultimately get their data, and then they work together to, the whole time, but then they're really analyzing, looking at that data as a full team. And then what happens is once that starts to occur, you really have the ability to make major discoveries, and you slowly move in to what would be a new research field. So this is where metabolomic epidemiology is a good example of how early on everybody started doing it was with multidisciplinary research, where everybody was kind of doing it and like looking at what everybody else was doing. Then all of a sudden, there was a point when certain consortia started and certain things started happening. I mean, those of you that know me and have worked with me, I'm thinking of COMETS. A lot of you were involved in a big consortium that was started at the National Institutes of Health. When that happened, all of a sudden, we're like, oh, we're all working together in a team. We're making discoveries with the full team, and all of a sudden, this new field has emerged. So I just want to, and if I forgot anybody on this, just yell out and say, you forgot me. But, aside from what we have for speakers today, we have a clinician, we have epidemiologists, we have a biochemist, two biochemists, and I guess I should have removed clinician from there because Deeba is sitting up here. But other disciplines for interdisciplinary team besides epidemiology and biochemists that were, in that figure, because that was primarily the biochemists and the epidemiologists. You have bioinformaticians, you have clinicians, you have chemists, you have data scientists, you have statisticians, and probably many more people depending on what that project looks like. So I'm going to put that figure up again, and what I'm going to say is this session is to talk about how you sort of started with multidisciplinary approaches to metabolomic epidemiology, ultimately found out and figured out and discovered how working in interdisciplinary teams is more effective, and that actually sort of began to establish the field. So in closing, I'm going to just highlight who's speaking today. We have Craig Wheelock from Karolinska. We have Deeba Husain from Harvard Medical School and Massachusetts Ioneer. We have Lening Guo from Precion Incorporated, and we have Jessica Lasky-Su from Brigham Women's and Harvard Medical School. So our speakers will go in that order: Craig, Deeba, Lening, and Jessica. So I'd like to bring Craig up onto the stage, and thank you all for joining us today.
Good morning. Fantastic to see so many people here, and I must admit to being very happy to be here myself. It took many days of travel to get here, so it's wonderful to finally arrive. And a second, they're jumping with my talk there. Excellent. So the hat I'll be wearing this morning is that of a biochemist, and I'll be trying to motivate to you what I feel is the importance of biochemistry in this multi and interdisciplinary paradigms that we've discussed this morning. So let's start off with some basic definitions. I like to sort of put everyone on the same page when I give a talk. So what is metabolomics? And at one level, we're at a metabolomics conference. We probably all have some idea of what this means and what our discipline is. But when I've pushed this definition a few times, I find that sort of in the details, there's not as much agreement as I would have thought. In the early days when I started in the field, I thought of metabolomics as analytical chemistry. We acquire our metabolite data, but there's many people working in this space who don't acquire their own data. They use collaborators or CROs, other services. They're still doing metabolomics. So how do we define this? And so I've listed a whole range of things up here. Skills and disciplines we'd see that be important in metabolomics. But what I would say is that metabolomics really is biochemistry. And if we think about this, metabolomics really is the omics science of biochemistry. It's a global approach to understanding the regulation of metabolomic profiling, or metabolic profiling, metabolic networks, and help us to understand the biological system. So this is a definition that I quite like because it gets to the fundamental of what everyone on this list has in common, is a desire to understand the biochemistry. And so the purpose of the talk today is to talk about how biochemistry can be a guide and help us, we can use it to make sense of our data, to understand it. And then to really interpret it, first we have to put our results within the context of the appropriate biochemical pathways. So it's necessary to understand the pathways and their respective reactions and how they function, which enables us to understand how metabolites might be perturbed from what's considered a normal range. Well, first you have to understand what's the normal range. If you look at your metabolite in a given system, what is the expected value? What is the range we should experience under normal physiological conditions for that system? And then we can understand what a perturbation would look like. And then if you want to start doing some sort of intervention to address the dysregulation of those metabolite levels, you have to understand the whole process first. And if you think about it, this is what biochemistry has been doing for the entire 20th century. There's an amazing body of research where huge numbers of people have worked to understand and catalog very precisely these metabolic reactions, understanding how substrate goes to product for massive amounts of metabolites. And so metabolomics is really well positioned to bring together all this information from the biochemical communities to help us address a number of the really fundamental questions that we have, not just in biomedicine, but across the board, in that understanding the biochemistry is really going to be key to understanding and interpreting our data. So biochemistry can be your guide. And I'm going to talk about a couple different areas to give you very specific examples to what I'm referring to. Pathway mapping, I think, is a good one. That's one thing that majority of metabolomic scientists do at some point as a way to understand their data. They put it in the appropriate pathway context. I'm going to talk a lot about isomers. This is one of the, I think, the glaring holes we have in much of the metabolomics that we do. Many of the methods don't account for isomerism. I've listed a few different types of isomers here. Endogenous levels, is the metabolite present in the matrix? Is it even there? Is it produced by that organism? Is it present at a concentration range that's high enough to be detected or quantified? Is it stable? And then lastly, some of the analytical biochemistry. There's a method of choice that you're using to acquire your data. Can it extract your compound? Can you detect it? Can you quantify it? Is it an appropriate method? And if you can use these different biochemical metrics, they can really help you to assess the quality of your data. Is what you're seeing, is it real? Does it make sense? At some point you have to take a step back and say, does this pass a bloody obvious test? This metabolite that I've annotated and reported, should it be here or not? So let's start off with pathway analysis. This is a summary of some work from Tim Ebel's group, which is really the main expert in this field. So I'd like to start off with making sort of the bold, broad statement that there's no such thing as global metabolomics or untargeted metabolomics. It doesn't exist. Every method is biased. We have no way to acquire the quote-unquote full metabolome. We don't even know what it is. We can't define it. We hear, David Wishart's latest talks, he's up at, 7 million metabolites in the human metabolome? I mean, the number keeps going higher and higher. I remember back in the days when it was 1,000, So nothing is global and unbiased. They're all biased. Doesn't mean they're wrong, but just you have to know what you're choosing and why you're choosing it. And so another way to look at it is know your blind spots. Is the metabolite you want to see, can that even be detected by the methods that you're choosing to apply in the matrix of choice? The pathway mapping efforts, they require some a priori biological knowledge of the pathways, and we assume that knowledge is correct. And I'm going to give you an example, very specific example though, of some mistakes that occur in these pathway mapping resources. Of course, there's species specificity in the pathways, and then is a really nice paper from Cecilia Wieder and Tim Ebel's group. They've shown that the number and proportion of metabolites that are detected in the pathway, of course, or in an experiment will bias those results. And particularly the annotations are extremely important in the observed significance of this. And so what they reported, and this is a figure from their recent paper, here's a citation. I highly encourage those of you interested to look at this excellent paper, where they've shown basically that misidentification of metabolites can have very profound effects upon the observed results of your pathway mapping. And that, of course, I mean, in any experiment that we do, there's going to be some level of misidentification. You know, if you're really going to try and do large-scale annotations, you know, there will be some mistakes. So what they did here is they simulated misidentifications for a whole range of data sets. That's what you see up here, different data sets that they examined. And they found that, you know, basically looking at false positives or false negatives in terms of the annotations, all of them had effects that were significantly above 0. In other words, that, you know, misidentification made some pathways suddenly significant, and some pathways that were significant lost significance based upon misidentifications. So how correct you are in your annotations can have a very profound effect upon the observed results. And then, as we said, that the assay of choice is going to bias what you're actually able to observe, detect, and eventually map. So this is a picture from the global KEGG atlas. And you can see that, you know, we've colored it, Tim's group has colored it based upon which type of analytical method you're applying to see which portion of biochemistry you can represent. But we see there's glaring holes. You know, I work in lipids, and you look up here, I mean, this is not well covered at all. And it's been, you know, well known in the field that lipidomics data are not well mapped on the KEGG. They don't have the right ontology structures to map the data on there. That's just the way it is, and so you need to know this. If you're trying to map your lipidomics data to KEGG, it's not going to work very well. This is data from our own group we published a few years ago, where we show the metabolite and the method specificity of mapping from our own in-house libraries. And so, you know, some pathways are well represented and some aren't. So when it comes to interpreting our data, we have to be very clear, you know, where we are on this graph when we talked about significance and coverage of a given pathway. Know your blind spots and know your biases. Now here's a very specific example from KEGG. So I work in lipids, specifically lipid mediators, chysanoids, oxylipins, and these compounds. So all of my examples from now on are going to be in this domain space. And the reason is not just because, you know, I like them and I want to share them with you, but because that's what I'm an expert in. That's my domain space. And the point of the talk today is to try and really stress the point. You need to speak with the domain experts to make sure that things are accurate. So this is the linoleic acid metabolism map from KEGG. And the way KEGG works, for those of you who aren't familiar with it, is there's a reference pathway which shows the relationship between substrates and products and the enzymes responsible for the conversion. And this comes up as you know, clear or white. So these are the compounds and the boxes are the enzymes, right? And then you can go ahead and you can, you know, color or annotate this for a whole range of organisms. So what I've done here is this is the Homo sapiens version. So the boxes in green are things that are present in Homo sapiens. So if you look at this compound here, so this conversion of these epomes to dihomes that you see here, what this is, there we go. For those of you not familiar with these compounds, linoleic acid is converted by P450s to the epoxides, which are then hydrolyzed by the epoxide hydrolase to the corresponding diols. So this has been known for at least 40 years. This enzyme, the SCH, has been cloned and crystallized and studied ad nauseum. So we're very, very certain of this pathway. We know it occurs in humans, but according to KEGG, it's not even fully annotated the enzyme, and it doesn't exist in humans. So clearly it's a misannotation. And even, laser pointer is not, there we go. Even dihomes is, you know, it's spelled wrong here. You know, so there's a problem in KEGG when it comes to this compound. Now, if you were not familiar with this pathway and you tried to map these compounds on here, it wouldn't show up for humans. Now, switching over to isomers, you know, chirality is something my lab works on a lot. I think it's fun. I like stereoisomers. So we do a lot of chiral chemistries. It can be really useful to understand the synthetic root of a compound. So I'm going to show you some data here. This is general oxylipin profiling that we performed. We did this in guinea pig airways. We saw a whole range of oxylipins that we detected. But to try and understand in more detail, what are these compounds? Where are they from? How are they produced? So we can look at the stereochemistry. So if we look at these chiral centers here on the monohydroxys, if they're racemic, so 50-50 R and S isomer, we know the source is autooxidation processes. But if we see that it is enriched in a single isomer, for example, the S isomer, we know it comes from an enzyme called a lipoxygenase, which produces exclusively the S isomer. So by looking at the chirality, we know how the compound is being produced. So we look at this compound from arachidonic acid here. This is the 5-HEAT shown in the structure here. We see fairly high levels of the compound being produced. So one would normally conclude 5-HEAT is there, so we see 5-lipoxygenase activity making a 5-HEAT compound. But if you look at the ratio, or not ratio, but the amounts of the S and the R, we see they're essentially equivalent. These are just different experiments we did. So it's a racemic production. So there's no lipoxygenase activity here whatsoever. But if you didn't have the chiral data, you wouldn't know that. Conversely, if you look at the DHA hydroxy compound here, this 14-hydroxy shown down here with the chiral center, we look at S and R, we see there's no R, it's all S being produced. So there we know it's lipoxygenase activity. So by looking at the chirality, we know how the compound is being formed. And if we didn't have this information, we'd risk making an incorrect assumption. And then the biochemical knowledge can help us spot incorrect annotations. So I'm going to give a few examples from the literature now. I haven't given any citations or references for this, because this is not a sort of blame, shame, and name exercise. So I don't want to put names on any of this. I hope it's no one's papers I've selected. Just don't say anything with your work. But the idea is just to show some of the consequences of what can happen from this. So this is data from a collaborator where they performed untargeted metabolomics. This is in mouse plasma, and they annotated their compounds. They annotated this peak here. And they said, okay, based upon the annotation, this is cortisol, common compound. We all observe it quite regularly in plasma. So they annotated it, they reported it, they wrote up their paper, they submitted it, and the reviewer came back and said, well, that's really nice data, but mice don't produce cortisol. Oops, all right? They had misannotated their compound. They didn't spend the time to look and say, wait a minute, is it the compound we're reporting, does it even exist in that organism? They just took the annotation. Now, I've shown the spectra here, where you can see that if you look closely at the spectrum, it's clearly misannotated. And the ion ratios are not going to match at all between the reference compound and the actual data. So it's clear it's misannotated. But it's easy to happen when you're dealing with hundreds or thousands of spectra in a given experiment. You can see how this could be missed. But when you're reporting compounds, does it make biochemical sense or not? Here's another example. This is more in my domain space. This is looking at urinary chysanoids in asthmatics. This paper reported the presence of this compound, this leukotriene D4. They're saying this was an important compound that was upregulated in the urine of asthmatics, which is fine, except it's not a urinary metabolite. We've run thousands of urine samples from asthmatics over the years. This is not a urinary metabolite. There's a different chysanoid metabolite from this pathway you see in urine. It's a misannotation, and this is published in a high-profile journal, in a supplemental. Here's a fun example. So this is getting a little bit specific for my field, but there's a lot of interest in fish oils, omega-3 fatty acids, and the potential health benefits of this. In particular, people have looked at the chysanoid analog. It's called specialized pro-resolving mediators, or SPMs. So this is the Cayman chemical pathway chart for the formation of these compounds. There's a fair amount of controversy in the field about the analytics of these, particularly that they're present at such low concentrations that I've seen conferences basically almost go to blows about publishing these data because analytics are, let's say, they're a concern. So let's take a compound out of here, for example, this resolvin E1 or RVE1 being produced from EPA. So here's some data to show you what this compound looks like. This is targeted data. So here's a full scan. And so we zoom in with targeted MSMS as an example. Oh, this is human plasma. This is our reference plasma in the lab. We look here. Here's this prostaglandin D2, which is a very low abundant dicysanoid. And here we see it at 140 picograms per mil. Then we zoom in a little bit more. There is a resolvin E1, all right? That peak. Let's not have a conversation about signal to noise. That's what part of the controversy in the field is about. But this is how they report RVE1, somewhere around 0.2 picogram per mil, right? Very low. But if you go to literature, you'll see a number of metabolomics, non-targeted global metabolomics, reporting these compounds. I don't believe it for a minute. This is 500 microliters of serum going through an SBE cleanup and concentration step, and this is the levels we're seeing. So if you're just going to be doing profiling without any concentration or cleanup, detecting these by non-targeted metabolomics, if you are reporting it, make sure extraordinary claims require extraordinary evidence. So if we go to the literature, here's a paper where they looked at mice who are given an omega-3 fatty acid diet. And we look at what they report. There's the 5-HEAT, and there's RVE1. So we saw, the questions about 5-HEAT. Not that it's wrong, but just that how's it being formed? You can't conclude it's lipoxygenase without the chirality data. RVE1, I have my concerns based upon what we just said. And then let's look at the next compound, this guy, prostaglandin H3. So I put the structure here, there. This is an endoperoxide with a half-life of about 15 minutes in plasma or in circulation. So I don't think you're detecting this by non-targeted metabolomics. It's not stable. And if you take the time to at least do a literature search in the compound, you'll quickly realize this is an unstable intermediate, and compounds shouldn't be reported in this matrix. Here's another fun example from my own group. So we're looking at lipid mediators in bronchial lavage fluid or bowel fluid from sarcoidosis patients. 135 compounds. It's put into a multivariate plot that you see here. We say, great, we can pull apart sarcoidosis in healthy individuals. And here are the loadings plot. And the postdoc working in this came back and said, there's really not so much of interest here. I said, well, why don't you annotate this? And so the top two compounds we've talked about already, the dihomes that are produced from linoleic acid by a soluble epoxy hydrolase. We know if we mapped that to KEGG for humans, it wouldn't show up because we already saw the enzyme wasn't there for this pathway, so there's a problem. But then the postdoc said, well, there's, none of the lipoxygenase products are present with a p-value less than 0.05, so it's not interesting. But if you look here, I said, wait a minute, every one of these compounds with an arrow on it is actually produced by that enzyme. So even though the statistics at univariate weren't pulling it out, the biochemical knowledge of the pathway shows that all of these compounds being shown in the loadings are actually coming from this enzyme. Maybe the study is underpowered. You can see that the actual magnitude is not so impressive here. But from a biochemical standpoint, it's all from the same enzyme. I find that very informative. So I see my time is running out. So how can we use biochemistry to understand our data? We saw the pathway enrichment analyses. They often provide very surface level interpretations. It's useful, but oxidative stress, inflammation, these are very high level ontology descriptions, and saying that inflammation is increased in the system, it's useful, but doesn't give us a whole lot of insight. And then we know that these can be affected by the coverage and accuracy of the pathway, as well as the accuracy of the annotation of the metabolites. So we need to be very, at least aware of what we're reporting. And that, most metabolomics methods don't distinguish between isomers, and this would be very important for interpreting your data. And as I said, the accuracy of the annotations can affect the significance of the reported pathways. And that, we can see an analytical bias in many of the compounds that we detect. We can think of things like phenylalanine and tryptophan. We always see these reported in metabolomics papers. They're high abundant. Analytically, they're very nice to work with. They ionize very well, and so we always see them, whereas conversely, some of these low abundance lipids, we're not going to see them. There's a bias in the way they perform in the instrument. So you really need to evaluate the potential to report a metabolite in a given matrix, organism, concentration, given time point. Does it make sense or not? And so oftentimes I feel this deeper biochemistry or biochemical knowledge and domain expertise is lacking, and it's very vital to have this to ensure the appropriate interpretations. So the basis for most diseases is found in faulty enzyme activity, improper substrate balances, faulty metabolic regulation. These are biochemical dysregulations. And all of these are affecting what's called normal metabolism. And so this is a real opportunity for metabolomics, which can give us a quantitative assessment of these pathways. But biochemical knowledge is really required for correct interpretation. So at the end, I say metabolomics truly is the science of biochemistry. And so use the biochemistry to help interpret your data appropriately. Thank you.
Okay, so that first talk was an example of why, when you have a metabolomic epidemiology team, you need a biochemist. Because otherwise you may have a situation where you misinterpret the data. Next, we will move on to Deeba Husain, who will talk about more of the clinical aspects and the clinical translation, moving forward, going from multidisciplinary to interdisciplinary, and then it will be followed up from the biochemist perspective for clinical translation of the biochemistry data, and that is Dr. Guo. And that will be talking more about multidisciplinary to interdisciplinary. And then we will top it off with Jessica speaking about a solid example of what interdisciplinary teams look like and what they look like for clinical translation. So you will see us going from the potential misinterpretations through how things are built to ultimately looking at what that interdisciplinary team looks like in practice. So I'm gonna give the floor to Dr. Husain.
Thank you. So I'm going to talk about clinicians' perspective on metabolomic studies. The goal of a clinician is to take best care of their patients. So from our point of view, we ask questions about what are the clinical challenges and how metabolomics can help us address those challenges. Most chronic diseases are multifactorial. That means they are caused by a combination of our genetics, the environmental factors, and lifestyle or exercise factors. Classic examples of these are cancers, diabetes, cardiovascular disease, many of the neurological degenerative diseases. The disease that I am most interested in is age-related macular degeneration because I'm an ophthalmologist who specializes in diseases of the retina. So age-related macular degeneration in its advanced forms causes loss of central vision. The picture here is what a normal person sees, and then in the advanced form of the macular degeneration, there is loss of central vision, inability to recognize faces and read and write, which leads to legal blindness. And this is the leading cause of blindness in the elderly in the developed world, and the third leading cause worldwide. And yet, it's frustrating to be a physician who's treating patients with macular degeneration because there are multiple unmet needs today in 2023. And this includes lack of accessible and reliable biofluid biomarkers, both for diagnosis as well as prognosis of this condition which is the leading cause of blindness. And what this means in practical life is that an older person needs to come in for a routine eye checkup to a specialized clinic for diagnosis, which becomes almost impossible because they have multiple other medical problems. They depend on others to bring them in, and that is the reason the diagnosis of this condition is missed in the early phases. It's only when disease is advanced and vision is lost is when people come in. Secondly, there is no treatment at this time for the early and the intermediate form of the disease, which constitutes about 90% of the problem. And most of this is because we have limited understanding of the pathogenesis of age-related macular degeneration. At the same time, being a retina specialist, I know that we have a lot of knowledge of genomics of macular degeneration. A very large international AMD consortium study has shown significant role of 34 loci and genes with both rare and burden for age-related macular degeneration. But the interactions among these genes and actually how they cause AMD is not fully understood. And therefore, we thought that molecular traits on a quantitative scale may be more associated with causal genes for this disease process and may be able to better contribute to the understanding of pathogenesis. That is where metabolomics appear to play a vital role because it's the end result of the genomic processes as well as the effect of environment. So it seemed like this would be one of the great ways of assessing any chronic disease, specifically age-related macular degeneration from our point of view. So this was a great idea, but as a clinician, I had no idea how we would actually do this study to look at establishing novel biomarkers and to understand pathophysiology of disease. I was lucky to be working in Boston, which has a very large metabolomics group with expertise in metabolomics, and I was very fortunate to collaborate with Jessica, who's an epidemiologist working in the field of metabolomics. And with her and Rachel Kelly, we worked for a year planning and sort of talking about the feasibility of the study, because I wanted to build a study ground up to actually recruit our own patients to be able to do it correctly from the start. So we, over a year, discussed study design, what samples we should be collecting from our patients, and we decided we were going to do it all. We were going to do plasma, urine, and tissue. And then we had multiple discussions with us and with biochemists in the group to discuss about what would be the most appropriate way of analysis. And I'm going to walk through this a little bit. So the study design we came up with was a prospectively designed study, which would be longitudinal. That means we would do yearly follow-up on our patients. We decided to recruit my patients from my clinic, as well as our collaborators' patients from Mass Sinai, the Mass General Hospital in Boston, and our collaborators and friends who are at the University of Coimbra in Portugal, to have two separate cohorts. And we were going to recruit all patients with macular degeneration in all stages of disease, as well as a control group of patients over the age of 50 without any age-related macular degeneration. And we did a complex study design where we were going to analyze the two cohorts separately, do meta-analysis, and whatever was the signal we found, we were going to do pathway analysis and curve analysis to actually talk about and know about the metabolites and age-related macular degeneration. In collaboration with the physicians in the group, along with the epidemiologists, along with Jessica's group, we decided that these are our patients, and we are going to collect as much information as will inform us about a multifactorial disease. So we collected information on them on medical history, which we actually created a standard form, so we will have the exact same information from every patient. We collected information on physical activity using the University of Washington RAPA form. We collected food frequency on a form which is made by the Harvard School of Public Health. All patients were our patients. We did dilated eye exam. And then we actually collected very detailed eye information, eye phenotype, by doing photographs on them, color photographs, and a new technology called OCT, which gives a cross-sectional imaging of all the layers of the eye, as well as we offered functional testing. So we collected a lot of information on our patients. And at the same time, we had a standard protocol for collecting early morning fasting blood and urine, which was to be processed within 30 minutes and frozen at -80. So at least the sample collection would be uniform throughout the cohort. We had multiple discussions. I don't think I need to go into this. Craig described this a lot. One of my collaborators actually did do an MR on a small portion of our study, but overall we decided that we were going to do non-targeted ultra-high performance liquid chromatography mass spec, which was run on the Metabolome platform for all of our cohort. So to get this study started, the first step was recruitment, and that involved actually working and hiring a group of clinical researchers, clinical trial coordinators, research assistants, and then to recruit from my clinic as well as other retinal physicians' clinic to enroll controls. We needed to enroll from our general ophthalmologist clinic. So it was kind of a collaborative effort at Mass Sinai to be able to recruit the patients we did from our study. And we had a parallel sort of group of clinical researchers and clinicians at the University of Coimbra that were involved with recruiting patients based on the protocols we had set up for this study. We recruited for over 2 years to be able to recruit 500 patients from both the clinics. And it was very exciting to find with the analysis that was done by our epidemiologist and biostatistician collaborators that we found that plasma metabolites are different in healthy patients versus patients with macular degeneration and also across stages of disease. And then the most significant metabolites were in the lipid and amino acid pathways. And up until now, there was very little information on AMD, so this was very exciting. And we got very encouraged to delve more into what metabolomics can tell us about macular degeneration. I have already talked about the fact that we knew a lot about genomics, but really how these genes function is something we did not understand. So we asked the question, can metabolomics actually help us understand the functional role of AMD risk genes? And for this, I collaborated with our statistical geneticist at the Harvard School of Public Health, who was actually one of the people in the original AMD consortium, and his postdocs, and they did MQTL analysis for us on our cohort, again, using the meta-analysis way of doing it, and found that we showed that genomic metabolomic associations support the role of LIPC gene and glycerophospholipids in age-related macular degeneration. We had collected urine on our samples, and up until now, we had not checked our urine samples. With the encouraging plasma results, we decided we were going to look at our urine samples. And unfortunately, in the initial analysis, we did not find any urinary metabolites that could differentiate between AMD and controls, but there was some signal, a low signal, when we looked at staging of disease. And to be able to better understand, we collaborated with Craig, who's an expert who's done a lot of work on urine metabolomics and is a biochemist that understands much better than us to be able to figure out what we were seeing in our data. And he explained that lipid metabolites actually are less likely to appear in the urine. And so far in all the plasma data, our most significant associations were with lipid. And maybe that is the reason we are not seeing a good signal. And maybe urine is not a good biomarker for age-related macular degeneration. But we are actually looking at some plasma surrogates in urine to see if that will be a better biomarker for AMD. The other question, which kind of most clinicians treating patients with macular degeneration are asking, that the current classification of disease is not what we want, because it's not based on pathophysiology. It's really based on clinical patterns, which is actually the truth for many clinical diseases. It's kind of like a pattern recognition, which is not pathobiology driven. So we asked a question that, can metabolomics help us with better classification of disease, which may be more pathobiology driven, and thus help in kind of finding early targets for disease. To do this, in collaboration with our epidemiologists, we thought of doing it in two ways. Either to look at the retinal imaging, so the actual features of macular degeneration, and seeing if those features are driven by certain metabolites, or the other way is the opposite way, to actually look from the metabolite side and see if there are any metaboendotypes that are correlated with clinical phenotypes. And this has been an interesting exercise that we are in the process of doing and publishing at this stage. So we've done extensive retinal imaging and correlated that with metabolomics, and actually found certain metabolites associated with certain specific retinal phenotypes, which would be interesting to look further to see if targeting those would help in treatment of early form of macular degeneration. And then Kevin, who's our postdoc, has been working on a machine-driven method called the SNF, which is a way of kind of doing metaboendotypes, seeing how within a certain cohort the metabolites cluster. It's a clustering method. And Jessica and her group have done it for asthma, and we decided to look at it in our AMD cohort, and actually found that our cohort had 4 endotypes which were driven by different metabolites. And we correlated this with our known AMD phenotypes and could actually have a severity among the cohorts. So this work is also in progress right now. We're making sense of it and putting it together in a paper. And the last part of it is that this is a longitudinal study where we are now actually in year 8 of collection, but we have finished the 5-year follow-up. And we have run metabolomics in different batches. Part of it was financial because we couldn't afford to run everything all in one to go. It's an expensive task to take. And part of it was that this is a longitudinal study. So we were collecting, we had 3-year follow-ups and we have 5-year follow-ups. So at the 3-year point, when we were starting to look at progression of disease to see if metabolomics was associated with progression of disease, we discovered that we had already run in 3 different batches. And we, before looking at differences and progression of disease, it was important to harmonize the data and merge the data. And we had many meetings with the biochemists and our epidemiologists and biostatisticians to have the best way of harmonizing the data to actually get the real values for progression of disease. And this involved merging the data and using specific matrix samples in the runs that we did to publish this. So at this time, we are continuing this exciting work to kind of use metabolomics to answer the difficult unanswered questions of age-related macular degeneration. Tissue metabolomics is the next step, which is already ongoing. All the work so far has been in biofluids and plasma and urine, which definitely has a role. But if we are going to talk about targets for treatment in the eye, we need to know what is happening to the metabolomics in the eye. And that is the stage we are at. With the pandemic, things were slow. This requires another big team of people to be able to do that, which includes biobank expertise, histopathologists to section the eye and image the eye, and then ways of figuring out how we're actually going to run the metabolomics on them. And the other project that is ongoing is actually to figure out the signals we have seen, are they going to be biomarkers for disease? And for this, we need to go to the next step of targeted metabolomics, for which we need another multidisciplinary team of biochemists and biostatisticians to help us run the targeted metabolomic assays. And both of these things are in the planning phase and ongoing. So I would like to summarize here by saying that clinically, there's a very significant relevance of metabolomic studies. It can help us better understand pathogenesis of disease. It can point to and help discover biomarkers for disease, both diagnosis as well as prognosis. It can give us more meaningful classification of disease, which may be more pathobiology driven and therefore actually point to early targets for treatment of disease. And therefore, it's important to, for ushering an era of precision medicine in chronic diseases, chronic multifactorial diseases. And this work is not possible without kind of many experts working together towards an interdisciplinary discovery. And I talked about the different types of people. So it really takes a village to be able to answer some simple questions, unanswered simple questions for various multifactorial diseases at this time, and requires multiple institutions to collaborate. And of course, everyone needs money to do. These studies are not inexpensive to do, so we need both government and foundation funding to be able to do this kind of work. Thank you. So Dr. Lane Guo, who's a biochemist, will follow.
Thank you. So good morning. It's a pleasure to be here. I think one of the goals, the main goals we do metabolomics is to hope one day we can apply our findings in the clinics for patient care. So having spent almost 20 years in the field of metabolomics, so few of us left our old jobs and put a little company together in North Carolina, and our focus is to try to build translational tools for metabolomics. And I can share some of our experience in those efforts. So just try to echo Craig's earlier point. There's really there's really no such thing as global untargeted metabolomics. The data is all biased in certain ways. So I think it's really necessary for us to understand the nature of the data before we're thinking about how to properly working on a translational effort of those studies. So we all understand from last decade or couple of decades, tremendous of data being published in the metabolomics as very significant findings. And in order to translate those results into clinics, we think it is necessary to take an intermediate step from your metabolomics data to build end clinical assay. So we call that this intermediate step as building essentially a targeted panel and as a validation tool, as a continuation of validation tool for the metabolomics results. So in terms of nature of metabolomics data, one thing we need to understand is there are still, even in a continued technology improvement in the last 10, 15 years, there are still significant coverage gaps. What I mean coverage gaps is important, biologically important metabolites. It's not in your data. So you're looking at what in the publication in the last 10 or so years, what's been published in terms of data set. We certainly come up a long way in terms of number of metabolites can be detected. In the earlier days, maybe we detect in many data sets 50 to 100 metabolites. And nowadays, with multiple platforms from different labs, and it is fairly common to see a data set roughly around 1,000 metabolites. Those are what I mean, the numbers are all annotated metabolites. So, and the data is, what I mean, the data is the coverage gaps is really created by the method you use, how the samples are extracted, and how libraries are created, if you actually can annotate the peaks in your data, and instrument sensitivity, so on and so forth. So every step of the way from the metabolomics data generation process, we left something out. So this is an example on the coverage gaps on a steroid hormone pathway. So the middle part is a classical biochemical pathway of steroid biosynthesis started from cholesterol. And I just simply pulled out two data sets, all recent publications, one out of Stanford, and the other is out of platform from Metabolome. And you'll see large part of the steroid pathway are not covered by either platforms because of the method setup and the instrument detection sensitivity. It all biased towards more abundant steroids. Some of the clinical important species are not really detected because they are in really such low abundance, and they are the metabolomics platform are not really sensitive to those metabolites. So that is one thing we need to keep in mind when we looking at our data and think about what to do next, is to say, if we find pathways that's important, we need to examine if there are significant coverage gaps. That means there's a possibility there will be a better, potentially could be a better biomarkers, but we don't know about it. It's not in our original data. And the other limitation is the quantitation of the untargeted metabolomics. We often hear the nature of the untargeted metabolomics quantitation as either relative quantitation or semi-quantitation. So that is all in comparison to the classical gold standard of clinical assays. It is anchored by two important factors. One is internal standard, so you can normalize the matrix effects and batch-to-batch variations, and many subtle differences from sample to sample that needs to be normalized with internal standard. The other is titration curves. So it has, for an absolute quantitation, we have to rely on titration curves with authentic chemical standard. All those are, in the majority of the platforms used today, it is actually quite challenging to do to meet those standards. So we end up the data, and especially for epidemiology studies, because we are often dealing with thousands to tens of thousands of samples, and also the data often needs to be bridged. And every time you bridge a data set, there are errors introduced, and the more you bridge, the larger error of the data you're going to have. So you may have to find a very statistically significant biomarkers, but we have to validate those findings to making sure they are holding up, the data holds up with targeted assays. So just the general considerations when we looking at our metabolomics data, thinking about designing a targeted panel for further validation. There are several important factors. One is we need to consider. One is the biological significance. It has to be data-driven, and the statistical analysis and pathway enrichment analysis is a big consideration. The other is the coverage. As I mentioned earlier, are we missing any really important metabolites, especially some of the metabolites may already been measured in clinical samples using our other technologies. So we need to take all those into consideration when we design a panel, design a targeted panel for creating a method. So just to give you a couple of case studies in our experience of taking this process from early discovery to a clinical tool. So this study is a collaboration of our company with a hospital consortium in China on developing a clinical test for a symptom called acute on chronic liver failure. So just a little bit background on these symptoms. So this happens, or acute on chronic liver failure, or ACL, or ACLF, happens to people with chronic liver disease, either hepatitis or cirrhosis. And some of those people could be stable for decades. But if there are any change in the condition, there's a precipitation event, often in the Eastern populations, it could due to viral reactivation. In the Western populations, often induced by alcohols. So those people could have basically a sudden liver decompensation, and all those people needs to be hospitalized. And those hospitalized people, roughly about 25 to 30% of them will be diagnosed as ACLF. So ACLF is a very serious condition, and all those people needs to be put in ICU for care. And 50% of them are going to die within 90 days, even in the ICU situations. So one of the unmet clinical need is to predict who's going to die, who's going to survive within this period. So the physicians can triage the patients. Perhaps they will be prioritized for liver transplant, or perhaps they needs to be treated very aggressively. So this is one of the goals the hospital group, the consortium wants to do, is to develop a tool that can use in the clinics. Essentially, when they have ACLF patients, looking at who's going to die, who's going to survive. On the other side of the coin, the 70% hospitalized people, they are not diagnosed with ACLF, but a significant portion of them, roughly about 20%, will develop ACLF within 3 months. So you could look at them as a false negative. So it will be very useful if we can predict who's going to be in those at-risk group. Then those people can be monitored and hospitalized very closely. So that's the goal of the study. And the consortium actually spent 3 years to build a substantial cohort, roughly about 5,000 people. It's the largest cohort in the world for ACLF. And they only had enough money to put roughly about 1,500 samples into metabolomics analysis. So the study design, we separate the cohort into half and half. Half is for discovery, the other half is blinded and kept it for later for validation of the diagnostic models. So we use the metabolome platform, and after we acquired the data, and we did statistical analysis and machine learning to prioritize the biomarkers, then design the biomarker panel, and we developed targeted assay on those metabolites, and eventually build a clinical diagnostic model for those two indications, essentially. So this is the process we've gone through. So if we're only looking at statistical analysis, we see about 270 metabolites with very strong differences associating with basically the survivors versus non-survivors. It is really not too surprising to see a big number that showed these differences, because liver really is a large contributor for the metabolism in the body. And when we see a dysfunction of the liver, it is really impacted in a very global manner. So we started with 270, and we have to restrict ourselves only looking at top 50 metabolites with the most significant differences between the survivors and non-survivors group. And after we remove him, because when we see a metabolomics data, many pathways and metabolites are often co-regulated together. So they are highly correlated together in terms of biological pathways and biological functions. So if we remove those co-regulating metabolites, they're redundant. and we end up with 25 biomarkers. Then we further looking at the availability of the standards, and both in terms of isotopic labeled standards and chemical standards, we end up with 15 metabolites. So we build a targeted assets for those 15 metabolites. And the table on the right-hand side is actually a comparison between the targeted assay data and the metabolomics platform data. So the best correlation coefficients we had was 0.7, and the lowest was 0.4, although all of those are significantly correlated. So it is really, it's an important point that if we only looking at untargeted data to predict disease, it may be quite different from what actually you could realistically have in the end. So in the end, we build two statistical algorithms. One is a combination of 3 metabolites. The other is a combination of two metabolites. The 3 metabolites combination can predict the survivors within 90 days with roughly about 0.85 AUC. That's the data in the test set, not in the discovery set. And we got even better results in terms of prediction of who's going to be at risk for ACL within 3 months. And with the AUC almost near 0.9. So in terms of timelines, the longest time for this project is on a sample collection. Took about 3 years. And the acquisition of metabolomics data, the data was actually done in China with a licensed platform from Metabolome. Took roughly a year because they have to get the sample on a queue. And it took us 3 months to build a targeted panel. Took another 3 months to run that targeted panel on those 1,500 metabolites. Then took about 2 or 3 months to do statistical analysis and finalize a diagnostic algorithm. So that's the timeline we had on this study. So just another quick examples on another tool that we built up for the gut microbiome research. And it's one of the hottest area in the last 10 years or so. And so this is a tool that's been used extensively for drug development by many of our collaborators and also being used on large cohort studies. So it's we curated all the biological important pathways from gut bacterial metabolism. The largest branch, of course, is the aromatic amino acids metabolism. Then you also see short-chain fatty acids, histidine metabolism, TMAO, secondary bile acids metabolism, and dietary compounds. It's a collection of nearly 80 metabolites, and we build a multiplex targeted assays, and we end up synthesizing roughly the isotopic standard for a third of those metabolites in order to make the assay work. So all 80 metabolites, the quantitation is based on isotope internal standard and 8-point calibration curves. So just to summarize, and we think metabolomics is a very powerful tool, but the data is not perfect. When we think about translation, we need to understand the nature of the data. And so those are the considerations in terms of building targeted panels, in terms of consideration of the biological importance, statistical results, and coverage gaps. So I think I'm going to end up here and pass it to Jessica to have examples on the clinical side.
Okay, I hope we're all hanging in there, right? So we've seen several examples here. We've seen Craig start with sort of illustrating how when we have these large epidemiological studies, if we don't have someone like a biochemist, it can be a disaster, right? Like, I don't understand how a lot of the data work, and there's been many times Craig has just looked at me and he's started laughing, and I'm like, what? And he's like, Jessica, that compound is so hard to measure, like, you can't trust it. And I think those are the types of things we all need to be aware of if we are epidemiologists. But I want to just take a step back and think about this idea of interdisciplinary research. Now, I'm looking around the crowd, and I see a few people here who are really experts in this interdisciplinary research idea. But I want to give an example of interdisciplinary metabolomic epidemiology from one specific paper. And we're going to go through it. We're not only going to go through it and say, okay, these people were involved. We're going to go through it and talk about the little comments that were made each step along the way that made something go from like a broad global metabolomic profiling to really a specific clinical recommendation. And so let's think about interdisciplinary collaborative research. In my perspective, and in this example, who is sitting at the table? Well, I want to talk about who is sitting at the table in the example of this Nature Medicine paper that came out about a year ago that several people in this room were co-authors on, and we want to think about this in the context of this case study. So really quickly, like, what was this case study about? It was about associating the metabolome with asthma. So that's the basis. But let's think about who was sitting at the table and who was key in this interdisciplinary framework. We had the epidemiologist, the analytical chemists, the programmers/statisticians, the clinicians, the biochemists, and let's not forget, if we're going all the way to clinical translation, the patient. And I will say in this case, the patient wasn't necessarily there all the way along the way, although actually, let me pause that. I have really, really bad asthma. My son has really, really bad asthma, so actually, I'm also the patient. Okay, so let's just talk about it. Now, we're going to give this example from the beginning coming in. In walks the epidemiologist, right? What do the epidemiologists bring to the table? They have these cohorts, they have biosamples, they have clinical data. What are some things that are important about what they bring in terms of broader understanding? They understand potential biases in the broader sense of the analyses of epidemiological observational cohorts. They have statistical knowledge and usually computational ability, and often know what it takes to make the proper inferences in the framework of epidemiology. And I'm wondering why that slide comes up there. But okay, so who came together here? Well, this is a really simple thing to start with. I was like, I have a cohort, I'm an epidemiologist, and I want to find other epidemiologists. So I contacted Claudia Landenberg, who is a colleague at the UK, who has some really large-based population cohorts. And I said, hey, I want to just look at the association of asthma and the metabolome. Can you use your cohort in addition to mine to do it? Very simple, basic framework. Okay, now as we're going to tell the story, I'm just going to talk about when come in important other people. So in come the analytical chemists. Now I'm going to say the analytical chemists may come in many frameworks. It may be that you're sending the data out for data acquisition. It may be someone you're working with closely. But what do the analytical chemists bring to the table? Well, number one, they have the equipment. They know how to run the mass spec and NMR. They understand data acquisition. They actually understand the spectral data. Remember Craig over here talking to you about resolving lipid mediators and how they're very, very hard to measure? Good example. I don't know that stuff. You know, they have established protocols and can recommend how best to obtain biosamples. They can identify what metabolites are reliably measured. They understand the strengths and weaknesses that you can see in the overall data that you have. And in all these examples, there's much more, but we're not going to go into those details because there's really too much there. I also want to highlight in this case, if you're working with a company and to have a data acquisition generated, there may be specific people within the company who have, who you're working with. And in this case, we did have that with Greg Michelotti. So coming on in this first case, right, what did we do? It was pretty basic. We had 2 cohorts. We had metabolomic data generated, in this case by Metabolon. We ran association studies just to identify associations with asthma. Now, one thing is we had really good power. So when we did this study, the first thing we saw was really, really strong associations, right? These associations were up to like 10 to the negative 20. Pretty profound, right? So this was something we looked at and we were like, whoa, this is pretty interesting. And not only did we see this in the discovery cohort, which we had as Claudia's because hers had the larger power, the larger number of samples, but when we looked at this, we saw the p-values in her study. These are like 10 to the what? I can't even see that now, but like they're 10 to the negative 10 or whatever. Now, our study, with this initial study of ours with asthma, is much smaller, but you see still very robust p-values in this replication, okay? So guess what? At this point, we completed the first part of what epidemiologists usually do. They find and replicate associations with asthma. So this is great. Check off the box. But let's think about this a little further. In walk the biochemists, okay? So who are the biochemists? And in all these cases, I've actually listed or shown pictures of people here who are biochemists who actually helped us along the way. You have Lining sitting here who actually the targeted steroid assay that they developed was something we worked on together for this project later. And you see here Nicole, who's sitting in the crowd here, and Amber Dolan, who are both people who've started working on this further, and Stacy Rinke, who really gave a lot of expertise. But the point is they understand these metabolic pathways. And one of the key things is when we look at this, the epidemiologists say, okay, we see associations. But the other thing we saw is, okay, we see that in all of these cases, these associations are inversely related to asthma. So that means reduced steroids is associated with being an asthma case. And, then this is somewhat obvious to even the epidemiologists who may not be the most bright in some of these areas. But when we look at this, we see, okay, all of the associations, literally 100% of these, are with steroids. And so that's when the biochemist comes in and they sort of look at this and, we say, well, let's talk about this and what we see here. And there's the whole discussion about, okay, well, really in this case, the entire steroid pathway seems to be reduced among asthma cases. Interesting and important. But in walk the clinicians, okay? Now, what do the clinicians bring to the table? They know the biology, they know, now this is really, really important, okay? As we see as we're going along, if we're going to clinical translation, clinicians are the key in terms of helping us make that happen. So they know the questions or the issues that are currently a main focus in the clinic. They know the existing tests. They know medications that are being used. They know sort of some of these underlying complexities with patient care, whether it's insurance or whatever. They understand the system. They understand the system to how to get what we do as epidemiologists to get actually implemented in the clinic. And they also understand, in this case with asthma, these patients are taking inhaled and oral corticosteroids. And this is something, of course, I also did know being someone in asthma, and that just goes to, right, this discussion. The first discussion, between the clinician and the epidemiologist of like, okay, we know that these associations could also be driven by the fact that asthma cases are taking inhaled steroids and oral corticosteroids. So you have the clinician perspective of like, yes, this is happening, and the epidemiologist. And my perspective, of course, is this perspective of, okay, this is confounding by indication. Is it that all of our cases, like the main reason that we're seeing this reduction is just because they're on inhaled steroids or oral corticosteroids? And talking also again to our biochemist, Craig here, who also, he's a biochemist and an analytical chemist, multiple hats in this framework. We also talked about the potential for oral steroids. In this case, we were able to cross that off. But the next thing we did is we said, okay, can we look at this and see how much of what we're seeing is driven by inhaled corticosteroids? And all we did was something really simple. We looked at the controls. We looked at asthma cases taking taking inhaled steroids, and we looked at asthma cases not taking them, right? So if you just do that, if the people who are taking inhaled steroids have a reduction, you can sort of get the sense that it's probably what is driving the association. So here you see, like, how many is that? Like 15 or whatever steroids. And in all of these cases, right, you see those asthma cases taking inhaled steroids have these lower levels, right? The asthma cases, now this is actually important, the asthma cases not taking inhaled steroids are usually the same as controls, but in many cases you can see they're slightly lower. Now this gets to important points when we're thinking about asthma physiology. So I want us to really put our heads and caps on, because thinking through this is super important. Because, okay, here, what do we see in this comparison of asthma cases not on any medication and controls? And when you look at this plot, you see that almost all of the associations still show a reduction in steroids. This is important, because this is actually fundamentally suggesting that potentially one of the driving forces for asthma and the physiology is fundamentally that they cannot produce steroids as efficiently as controls. Physiologically, that is a very important thing to understand. But what else do we see here? We also see that asthma cases taking steroids versus those who are not are still lower. That's also important. A lot of what we're seeing could be driven by this inhaled steroid use. Okay, so we've thought through a lot of this. We see potential effects of inhaled steroids, non-inhaled steroids. In talk comes a clinician. Now, by the way, this is my mentor of about 20 years. His name's Scott Weiss. This guy has an H index maybe over like close to 200. One of the leading epidemiologists in asthma, and probably one of the foremost genetic epidemiologists in asthma in the world. And he is a great mentor. And, he sits down with me, and, we're in our meeting. And one of the things we talk about is, well, there's this really important thing that's happened over the last 40 years in asthma. Since inhaled steroids have come on the market, right, there's been, there's a lot of things we know they do. We know these steroids can reduce growth if they're given in kids. So kids may not reach their optimal growth. I was meant to be 5'5". Can I tell you, I really wish I'd gotten there, but no. But fundamentally, there's other things that they have always sort of hypothesized and surmised, but never been able to prove. And one of them is, while there is this premise that these inhaled steroids stay in the lungs, there is a thought that there are much larger systemic effects, but hopefully not as bad as like oral steroids. And the fact of the matter is, these have come on the market in the 80s. It is now, 40 years later, and we really don't know how much of this is causing potential adrenal suppression. So further reducing the functioning of your steroids in your lungs. So Scott looks to me and he says, hey, Jessica, what would really be helpful is if you can demonstrate that there's a clinically relevant adrenal suppression. Okay, now I want us to think about these words, clinically relevant adrenal suppression from asthma use. And so I said, oh, sure, I can do that. I'm just going to tell you the epidemiologist's understanding of clinically relevant. Just put in your mind what you think clinically relevant is, okay? What's clinically relevant? So this is what I said. I said, okay, now I have this whole step towards precision medicine. Lasky-Su steps towards precision medicine. What do we want to do? We want to make sure this is an effect, and then we want to, see if we could quantify it and then evaluate the clinical impact. And I'm thinking I know what the clinical impact is. You think about what could be, how could you show there's a clinical impact of potential reduction in steroids? And we'll get to it. So let's go through what we need to do to actually prove that in a larger framework. So, in order to actually show that this is a real effect, right, the best study design is a randomized controlled trial of ICS use in asthma. And then you look at those steroid levels in the asthma individuals on the ICS and off, the best study design. It just so happened that in our lab, we actually have probably the best randomized controlled trial for ICS use in the world. And so I already had that data that we had, measured on metabolomics. In talking about this, I want to actually highlight Craig here because even though he comes in as an analytical chemist, he is also thinking about the same idea and talking to us about what we should be using in thinking about this. But we had fortunately already had this randomized controlled trial done at the Broad. So we take Clary's data that he has, and we look at this in the context of a strict clinical trial, 4 years on this meds. And what do we see? Now, there wasn't as many steroids measured on this, so I just want to highlight this platform at the time only had cortisone and cortisol. But in 4 years on a randomized trial, we saw significant reductions in risk. Now, this is pretty important because this clinical trial had really, really low doses of inhaled steroids. So we're showing that we can see a reduction in risk with low-dose steroid use in a clinical trial. So this is important, so we have the next step. Now, my vision of this has really changed. The first thing was just associations with asthma for metabolomics. And now we're like, can we actually show that we see this reduction in steroid function on inhaled steroids? And we do the next step in this randomized controlled trial, and yes, we see reductions. So we can check off a box of we've interrogated this in an R.C.D. So this is great, and this is important, but we did this using untargeted metabolomics. And really, if we want to get to this step of clinical translation, what do we need to do? We need to actually quantify these steroids and really look at this from the framework of what a clinician would do. Now, this was an interesting point in the query because in comes the clinician who says, hey, Jessica, I want you to know, there's already a test that exists in the EMR to quantify adrenal suppression. It's called ACTH stim testing. So we could actually quantify it within the current clinical, in current clinical measures. Now, this is an important thing because typically the step from interrogating an RCT to quantifying the effect takes, someone like Linning who can create the biomarkers, and then you measure them, and then you show it's clinically relevant. We were able to jump over that because the test already exists, guys. The question, though, is, can we get the data to actually see if we can see this effect? So this is when Scott says to me, hey, Jessica, well, you can look at this if you look in the electronic medical records. Well, fortunately for us, we have access to the Mass General Brigham electronic medical records, which captures about 4 million people within Massachusetts, eastern Massachusetts, and we have IRB access to actually look at that data. But there's a problem. To quantify those effects, the data are a mess, right? These ACTH stim tests are not common. They're done in all of these different labs throughout eastern Massachusetts that need to be curated together. So in come the next people, which are the programmers. And what do the programmers bring to the table? Well, the programmers bring to the table the fact that they can deal with really messy electronic medical record data, and they can grab unstructured data and they can curate it and actually help us look at it in a framework where we can assess whether we see this reduction in steroids using this formal clinical test among asthma cases taking ICS. So that forms the fourth part of the study design now, where we extracted these ACTH stim tests from the electronic medical records, and then we looked at asthma cases on ICS. Now, I want you to actually look at this because not only, sorry, my thing isn't working here. Now, if you see the 4 lines here, not only do we see that asthma cases have this lower level of steroids, the other things that were pointed out by expertise in endocrinologists who came in, so other MDs who talked about this, and talking with our other biochemists, that when we're looking at this ACTH stim test with cortisol, there's this diurnal variation in it. So we couldn't just look at these levels at one time point. We actually had to look at when each person's blood test was taken and map it over time. And what was amazing in this is in a 24-hour period, you can see it doesn't matter when. These asthma cases had really low levels of inhaled corticosteroids. So this is very clinically relevant, right? These levels are low. So there is a clinically important drop. And this is where I looked back to my mentor and I was like, we found it. We found the clinically relevant difference. We found that there's this clinically important drop in cortisols. Yay, we're done. And that's when, when we looked at this idea of evaluating the clinical impact, we had my mentor come back to me and say, no. That's not the clinical impact I was talking about. You showed that there were lower levels in asthma cases. But what I want is to ask these questions. Well, number one, I think it's important if there's a dose effect, because clinically, that's going to be a major thing in how we would treat this. But when I say clinically relevant, I mean, are these patients having side effects? Are they coming in? Are there things that aren't being treated in these people because they have low levels? That's clinically relevant. And that's when it hit me that I'm looking at this to try to prove the level's low. He's looking at this from the patient perspective, who's coming in and saying, how can we optimize their health? That's a difference in perspective. So then what did this require? I just want to highlight this in terms of this multidisciplinary, interdisciplinary aspect. When he asked these questions, which were also asked by the journal, of course, then we needed to go back to the electronic medical records, curate that data, and answer these questions. And some of the things we found that were very important is, number one, there was absolutely a dose effect, and it didn't matter how we looked at this. If we looked at this as we're looking at the actual MCGs. We see that with increasing MCGs, there's a strong decrease in steroid levels. But the other thing down here that we did is we actually looked within one specific type of asthma, fluticasone, in type of medicine, and we looked in the categories and saw, yes, with increasing dose, you definitely see these reductions in risk. But then more importantly than that, when we went and we looked at, are there actual clinical symptoms? Because, the thing that's hard is some of these are so aren't things that a patient would necessarily associate with consistent inhaled steroids. And this is where we see there's a pretty profound increased fatigue, and there's pretty profound increase anemia, and these are increase of over twofold increase in the associations of these pretty important side effects. So now, yes, we are evaluating important clinical impact. But I want to go back to the last person on the table, because when we had this paper submitted, the reviews were actually some of the best reviews I've ever had in my life, and they was from Nature Medicine. So I was really pleased about that. But one of the things they really pushed on us was, what is the clinical relevance and the clinical recommendation? And I want to go back to this last person on the table, because this last person on the table, we have to remember, if we're going to clinical translation, it's about the patient. It's not about the cortisol levels, it's about the patient. And actually, this is a picture of my son, from ages 4 to 8. And I want to show you, this illustrates how, steroids have dramatically helped his health. And I know that inhaled steroids have had a tremendous public health improvement on asthma. So when thinking about this idea of the clinical recommendation, it's not about stopping inhaled steroid use. That's not it. But in thinking about this, I was thinking about this, and this is where it comes to this interdisciplinary, but also just the collaborative team that you have. I was on a call with a pediatrician who wasn't really a part of this other project, but he came in and we were talking about this, got on the topic, and he said to me, Jessica, when I am treating patients, he's like, this is really important what you found. When I'm treating patients, what happens is if a kid comes in with asthma and they have, let's say, a bacterial infection and they're having trouble breathing, I always put them on inhaled steroids. And frankly, I probably put them on too much for too long. And he said, it would be really easy to reduce the dose and reduce the length of time. and that is a really important thing. And in fact, in looking at this, one of the things we found was you could reduce the dose in half while maintaining 93% efficacy. Now that's dramatic. If you can reduce the dose by half, you're reducing, this risk dramatically, and you're maintaining efficacy. And so really when it came to the recommendations, it's about monitoring these doses and monitoring these levels. Now, ironically, when this paper came out, it came out actually sort of simultaneously to the global initiative of asthma, changing their recommendation to saying that inhaled steroids should be the main source of reliever medication. So they're really recommending taking out bronchodilators, which has sort of been the mainstay for the last 50 years, and putting in inhaled steroids among all these people. And this was interesting because really I'd say that like our paper would probably really argue against that. And it was coming out pretty much right after this was released. So this also became something we highlighted in the paper as something like, hey, this just changed, but we need to think about this and potentially change and have the bronchodilators stay as the main source of reliever exacerbations. So in this case, we went from this global cohort discovery, and I wasn't really anticipating this, but through these subsequent studies, we went all the way to sort of, I think, what are important clinical recommendations for asthma. But in this process, when we think through all the people that were involved and we look at this table, we see, okay, in come the epidemiologists, the analytical chemists, other bioinformaticians, the clinicians many places, people who act as analytical chemists and biochemists, and other programmers and more epidemiologists and more programmers and statisticians, other clinicians, talking about specific aspects of treatment, other analytical chemists talking about specific things like in the libraries, and this continues, and finally we get to this point where we have the patient as well. And this, my friends, is sort of the vision we have for what interdisciplinary research is all about. And I'm going to stop there because really this is my acknowledgement slide, and we're now going to move to the discussion panel where we're really going to be talking about and fleshing out how we can do this better as a community. And so I'm going to hand it back to Krista to have that discussion.
Okay, so now you've heard some various examples of what happens when you don't include people in the interdisciplinary team. You've seen how some of it goes from multidisciplinary to interdisciplinary, and then you've shown a clear example of what the interdisciplinary team looks like in epidemiology. So I'm going to open up the floor for questions for our speakers. There's a mic here and a mic here. If you could just get up to the microphones. I'm sorry, there doesn't seem to be one in this row, but if you can make it over to this one. So okay, I'm going to start with this gentleman here over in the blue. Oh, thank you, Leo.
Well, this is excellent. I just want to go back to step 0 of all this, which is your patient selection, study design, understanding of your inclusion-exclusion criteria. Without really saying anything sacrilegious to clinical trials, have you encountered an example where you loop back in understanding of disease pathway in order to better design your study, knowing what to look for, what patients to enroll, what inclusion-exclusion criteria, comorbidities to take a look at? I mean, I would argue if you are able to deliver robust diagnosis in a subpopulation of your cohort, that's still going to be valuable in terms of clinical diagnosis rather than, kicking the can of that p-values down at the population level.
Who wants to take that?
That's a great question. I don't know if this is on. Can you hear me?
Yes.
I'll start, but then I think I'll pass it on. I think what happens, at least in terms of how this works, is each set of the way, right, when you do a study, you learn more that informs then how you might select the next study. And I'll give you one example. In actually the very first collaboration I had with Craig, we were studying asthma patients. And one of the things he said, when we were talking about how we're going to select them, was to exclude smokers. And I think that was one clear example that I think was really important because smoking often, has this other comorbidity with starting COPD. There's a lot of complications when then looking at that in the metabolome. And so I do think as we go along, one study informs future studies of how we do it.
And kind of when you're doing disease focus, like we did our study, the way we did it was to get, we know what the unmet needs were and what we were trying to address. So we tried to get as much information on this cohort because we had the advantage of designing the study from scratch. And these were our patients. So we were, they were coming for their clinical visits as well. So we were getting as much information as possible on all the possible roles of multifactorial diseases and ruling out any other retinal disease. So the controls were patients who did not have any other retinal disease. And again, we had the advantage because we were starting from scratch and we knew these patients of actually screening them ahead of time. Even the AMD patients did not have any other retinal disease, neither did the control. So that way, we were reducing that kind of bias on them. That was the advantage of planning ahead of time. That is not easy to do when you're collecting data which is already there. And that's part of the reason we decided to take this route to be able to get the most clean data based on what we know about pathogenesis of disease. Diet information, which we are in the process of looking at, retinal structure, retinal function, to see how metabolomics can explain that parts which we don't fully understand.
Any additional comments from the panel, or can we move on to the next question? Okay, Phil?
Presentation by the whole groups here. Jessica, specifically, a couple of questions of interest. Has there ever been any kind of anecdotal evidence of association of prolonged use of corticosteroids and fatigue, like case reports and such that made it?
Yeah, so the interesting thing is if you look at the literature, what's amazing is there's a lot of like anecdotal or small cohorts, but what's happened among a lot of pharma is they want to show either that there is an effect or this or that. So you look at so many of what's been out there as RCTs, and there are things like randomized controlled trials over 4 weeks. Sure. Yeah. And so like over 4 weeks, you can't tell stuff, you can't really differentiate these. I have been on inhaled steroids for 40 years. 40 years, I am 100% sure I have adrenal suppression, right? And so that's the problem is right now, they have a lot of anecdotal stuff, and you'll see probably 50 papers out there, and they have very clear evidence of reduced growth, but they don't, there hasn't been studies over this very, very long term, and that's the issue.
Is it known you have to typically up the dosage because of some kind of tolerance? Or that's typical?
So there is this whole thing of steroid resistance, which is another entire area, because if you're taking this for so long, what can happen is people who have asthma, right, they have this pro-Th2 inflammatory response. When you then are taking steroids and you're getting steroid resistance, it further exacerbates some of this Th2 inflammatory response that's already out of whack, which then makes it much more difficult to treat, right? Because then you have to either increase the steroids, and there are not a lot of other great options. I mean, those are some of the problems. I don't know if you have.
I'll just briefly add to that steroid resistance is also one of the key symptoms of the severe asthma. And so there's a whole subphenotype we're dealing with of these severe asthmatics that are not able to modulate their disease with either ICS or OCS. So there's clearly a different mechanism going beyond that. We think actually a lot of that is non-T2 driven.
Yeah.
Maybe last, and maybe a big question is why the increase in prevalence of asthma and other autoimmune disorders?
Why do you what?
Why is there the increase in prevalence,
Why is increasing prevalence of asthma?
Yeah.
There's a lot to unpack
can we
in that
prevent it?
in this panel, but I would say a lot of it has to do with air quality issues, to be honest.
I think one thing we're going to actually see, I think there are a couple of things I'd say on that. I think some of it is environmental exposure, but changes in environmental exposures interacting with genes. And another key thing is, I think, potential change in sort of viral exposure, because I think that is probably going to be also an important one of these environmental things. It's my personal thing.
Okay, back with the mic in the white shirt, please.
Thanks a bunch for a really nice set of talks. So we heard about a number of different diseases, AMD, asthma. Can you give any insights for how you would pick diseases where metabolomics might yield high clinical insights compared to something like transcriptomics or genomics?
Who wants to take a stab at it?
I mean, I think multifactorial disease is the first basic thing, and metabolomics is, as I was showing, is kind of downstream, and that nicely captures the effect of the genetics and the proteomics as well as the environmental effect. So most multifactorial diseases, I think metabolomics will inform us in a very good way.
I think one thing I'll say is, starting out as a genetic epidemiologist, there's a lot we've seen with, there's billions of dollars of research that's gone into genetic epi, and for chronic, complex diseases, really for the majority of them, what it comes down to is, polygenic risk scores, because any individual genetic variant has a small effect size. I think one of the potential promises of metabolomics, for multifactorial diseases is the effect sizes are larger, right? And I mean, I think lots of us can speak to that, and that, that seems to be a potentially seemingly important thing that we're observing relatively consistently. My issue, though, is going from general associations, like these are general inflammatory associations we see, over all chronic diseases to specific, markers for particular diseases.
Any additional comments?
So just my experience, metabolomics is a very broad tool, and it can be applied to many diseases. But one area I think we have not seen many successful examples is early cancer detection. So I think the reason being, I'm just guessing here, is cancer is just simply a reprogramming of the existing metabolism. For the early cancer, when there's stage 1 or even earlier, the impact of the tumor on your blood chemistry is very, very minor.
Okay. All right. Rima? Oh,
I missed the first one. But general comments about people who are using metabolomics in large clinical studies, epidemiological studies, biobank samples. Number one, many of the diseases we look at are quite heterogeneous. Many of the diseases that we look at do have common mechanisms. There are no boundaries. At least when we study CNS disorders, there's a myth around putting these boundaries. This is depression, schizophrenia, Parkinson. Nonsense. Biochemically, there's a lot of relatedness. Inflammation, immune dysregulation, common mechanisms across these disorders. The exposome contributes in profound ways to everything we are looking at, clearly and loudly. Right? So in thinking about and in leveraging these epidemiological studies from filling in gaps of biochemistry, that is robust, reliable, that other people can use. I hope that we can all make our data public, as we are with NIA, where we are making data public in real time. We produce data, we upload it, we analyze it along with everyone else in the universe. As a metabolomics community, we at least need to aim to make everything public, in the public eye. I think that is where we advance a field. Thinking about these complexities with a continuous dialogue, I think is going to get us there, but the complexities are many. We realize it. The complexities are many. Yep. I just hope we can continue the dialogue.
Does anybody want to respond
You
to
know, and
that?
I think one thing that you said that is incredibly important is, you know, I was talking about the specificity of disease, but your point, and you know, there's some beautiful papers on that. I know that came out, the comorbidity paper about a couple of years ago with Claudia and Gabby and a bunch of people about, you know, the overall comorbidity we see. And it's tremendous. And that is actually really important to capture. And I think one of the things, putting this to what Craig said earlier and what you commented on, that, you know, metabolomics in some ways essentially is biochemistry. And some of the general inflammatory responses we see that are comorbid, that biochemistry could be very important to translate to other areas of science or other omics or this or that those general changes in biochemistry are something fundamentally important that could help many people in research if we had biochemists correctly annotate it and deliver that.
Okay.
NIH institutes are coming together to think about inflammation, immune dysregulation as a possible target, as an inducer of pathogenesis across diseases, and the idea of thinking a paradigm. We do not want to target the symptoms of disease. We want to target the underlying mechanisms.
Yeah.
And some of these mechanisms are common. So where do we go from here? If they are in common, we're not going to put them on the side and say this is inflammation. On the contrary, we're going to put them separate and think about it in a different way.
I mean, I agree completely, and that goes to a lot of what we're trying to do here with metabolomics. at
Craig,
least,
could
you
you
know.
just repeat the main point? Because I don't think everybody heard it,
Oh,
just the headline of it,
Okay.
if you could. And then we'll move on to Fred after you answer, because I want to make sure he gets to answer his question. But can you just headline that so everybody knows what was said?
I'm trying to, I'm jet lagged, I don't remember anymore.
Sorry, Craig.
Yeah, go ahead, Rima.
Thank you, Rima.
Oh, she's going to headline it. Headline, Rima.
Thank you.
You know, I mentioned these common mechanisms across disorders. The boundaries are diffuse. Inflammation, immune dysregulation is one example, right, which could be underlying mechanisms across many diseases. A new paradigm of thinking, and within large initiatives within NIH, is to say, look, we are going to look across diseases now, Alzheimer's, Parkinson's, diabetes, autoimmune, and what have you, and refocus on some of these common mechanisms to start to treat underlying pathology instead of symptoms.
Right, exactly. I mean, the idea is that, you know, thinking more as a metabolomic scientist or as a chemist is that we can restructure the way that we define things. And I work within the space of asthma, and the goal of what we do is to move away from clinical descriptions of the phenotypes to molecular descriptions of it. So we actually have, you know, quantitative molecular-based descriptors of different disease domains that will be more informative because there's an idea, there's a treatable endotype behind it that can be described by the biochemistry versus sort of overarching nondescript clinical symptom-based things. So it's trying to get more to the direct biochemical molecular descriptors of it. And those are the mechanisms that we can target. And of course, that will be, you know, maybe not redundancy is quite the right word, but we'll see the similar mechanisms that'll be taking place across multiple pathologies, and that'll give us a lot of information on how we can actually treat different syndromes.
The general comment or point that you've made, though, that I think is incredibly important is whether you call it the overall allostatic load or the overall comorbidity that has these common underlying mechanisms of increased inflammation and dysregulated immune function as you age. That being a major target, I think, is incredibly important. Yeah.
Thank you. We're going to make sure Fred gets to ask his question. Hey, Fred.
Thank you very much. This is a great set of talks, and Jessica really brought it around full circle. And I think that Jessica's talk really can begin to provide a great framework. If we can have examples like that, we can build a framework from preclinical studies to clinical recommendations. And I think that's really great, Jessica. I don't know how long it took, you know, to have all of that accomplished, but it's really great. My question is about the misidentification that Craig highlighted, and Lining echoed that in his talk. And I'm just wondering, as an epidemiologist, when I get the data, the metabolomics data from the lab, how concerned should I be about misidentification? I know that no experiments are perfect, but have you tried like quantifying the prevalence of misidentified metabolites to kind of guide the community? As an epidemiologist, when I get the data from the lab, I assume that I'm most interested in the named metabolites, and and I assume that the data are correct, and I proceed to, the best I can do is make some decisions based on the QC data that accompanies the data from the lab. So I'm just wondering if that is, how concerned should I as, being on this or that end. The clinical, the biochemist collaborator that I would have probably is the one who produced the data for me, that we work on the study together. And so
it's a fantastic question, and it's a lot to answer. And so to keep it really succinct, I would say that in many ways it kind of justifies the reason we have this conference, is that you can't be expected to do everything on your own. So we try and talk about in workshops like this, what are the important metrics? What should you be using as a way to assess the data? And so we can tell you these things. And so whoever is your data provider in your instance, at least you know what questions to ask them. You say, okay, so you know the difference between an annotated metabolite, whether it's annotated with a database match or whether it's annotated with an actual analytical standard. You understand that? You can ask that question. You can say, how many of these metabolites are standard confirmed? So whoever's providing your data should be able to give you some basic QC description of their data analysis or annotation pipeline. And that's what you'll have to use to assess what you feel is the quality of the annotations of what you're being provided. And that's going to be very specific upon whichever lab that you're working with. In terms of the specifics, the little tiny points I was referring to, because they're very specific examples, those are only going to come out when you work with a domain expert. You're not going to be able to go into that level of detail with a large data set of thousands of metabolites or tens of thousands of features. It's not realistic. But as you coalesce your analysis and you focus on very specific pathways that are of interest to whatever question you're asking, at that point, then it is very important to engage with an appropriate biochemical person who can check those things for you at that point. I certainly wouldn't go to publication not having a clear understanding of the biochemical pathways that are being reported along the lines of the metrics I reported in my talk.
Thanks, Craig. I think we only have time for one more question, and we had the mic over here first, but hold on. But I want to make sure you get your question answered. We just don't want to keep over lunch, so can you please make sure you come up at the end and get your question answered? The speakers will be happy to answer it. I just know the only thing standing between you and your lunch is me, so I want to be cautious. So we're going to have one last question with Sandy, and then we're going to wrap the session up.
Yep. Thank you for the panel. It's really an eye-opening session. My quick methodological question for Jessica and Craig. Is there a way to adjust for bias from compliance? And following that, maybe is the metabolomic data, can it help actually have a signal of those who are compliant with taking the inhaled corticosteroids and those who are not? Can you comment on that?
So I'll say one thing about that. The one thing with compliance, so let's say in the RCT, what's the compliance of people on the ICS and not? The most important thing, and this is actually a broad thing to remember when you're looking at something and you want to look at the effect of a medication versus non, you're always going to have non-compliance. That is part of it. The thing you want to remember, though, is that non-compliance is biasing your associations toward the null. So if you still see an effect, it means despite the non-compliance, there's an association. And so I think when you're looking at results or whatever, and you're looking at something that's a binary variable, if it's a bias toward the null, that always is okay if you're seeing something, because it just means you're seeing it despite non-compliance.
Do you have a quick comment?
I don't want to keep you away from your 12:30 mark, but I would just say very quickly in 30 seconds is that I mean, yes, we are looking for metabolic markers of compliance with both ICS and OCS, and I think it's very important for a whole level of reasons of the effects that these medications have upon metabolism. And I would take it one step further, is that we need metabolic markers of multiple medications. And so anytime we do a metabolomic study, we always stratify for whatever the common medications we're seeing within the given group, because it can have very profound effects upon the observed metabotypes.
Great, thanks, Craig. Well, I think it's time to wrap this session up, so I want to thank our speakers, and please just hang for a minute so this gentleman can get his question answered. We want to make sure he gets it answered, and I want to thank our speakers. I want to thank Leo, who's always the most helpful person in the room. Thank you, Leo. And I want to thank all of you for joining us and sticking with us till the very end. We're very excited, and we're looking forward to seeing you all at the opening session, and I hope you all have a great conference.